Good morning. First, I would like to thank Alvaro and Hanneke for the invitation to participate in this symposium. Today, I will be talking about work done at TU Delft with a former postdoc, John Buchner, and two PhD students, Koen Muller and Junaid Mehmood. So the microswimmers that I will be discussing now are somewhat different from the ones that you've heard in the previous talks. These are biological microalgae. And so this symposium is about the physics of these microswimmers. And of course, in this case, they are important in ecology, but also of growing interest in a variety of industrial applications involving the large-scale production of microalgae for the food or the cosmetic and pharmaceutical industry, also more recently for new biomaterials and in particular bioplastics, which you can 3D print. And even for new living photosynthetic textiles and many other applications. Now, if you consider these suspensions of motile microswimmers, a question of fundamental interest is how the dynamics of the motility at the microscales translates and affect the microscales. And in order to get insight into this question, we use a unique experimental setup which we have in the lab of aerodynamics and hydrodynamics in Delft, which is a 4-camera view microscope. We place in a flow chamber here a suspension of motile microalgae, in our case Chlamydomonas, and we illuminate our sample with a red LED light source. We record the motion of the cell simultaneously with 4 SCMOS cameras by collecting the forward scattered light, and this is essentially performing dark field microscopy. All of the cameras view the suspension through a single large objective, and each camera with an offset angle from the axis of the objective, such that all cameras view the common measurement domain, but from 4 different viewing directions. We use an in-house tracking algorithm, which was developed by Koen Muller, our PhD student, in which we first track each cell in 2 dimensions in the image plane of each camera, before linking and triangulating these into 3D tracks. With that, we can track all of the cells in the suspension throughout the entire depth of the flow chamber, which is about 2 millimeters. So one of the things that we can look at and that we are currently looking at with this experimental setup is the interaction between 2 swimmers. Now, as you can see here, the suspension is dilute, such that we can follow isolated interactions between a pair of microswimmers from 4 different views and see how they interact. And what we try to do is to characterize the nature of the physics of the interactions. This is ongoing work by a PhD student, Junaid Mehmood, but today I will be talking about something slightly different. I'll be mostly focusing about another problem, which is the interactions between cell motility and solid surfaces, and in particular planar surfaces, which is an important problem related to surface colonization, biofilm growth, and biofouling. This work has been carried by John Buchner, who has just started a faculty position. In these experiments, we record our suspensions, and we find that the cells, the Chlamydomonas cells, tend to accumulate at the surface. This effect, surface accumulation, has also been observed for bacteria. Here, we plot the cell density as a function of the distance to the wall. And we find a significant increase in the cell density at the surface compared to the density in the bulk. Here we normalize the density by the density in the bulk. The density here is the important microscopic quantity, and the underlying question is, can we link the physics of the microscale motility of this small scale? In that case, this Chlamydomonas cell is about 10 microns in diameter and swims about 100 microns per second. And so can we link this to the relevant microscale quantity, such as the cell density and surface accumulation? which are the driving quantities behind surface colonization. To address this question, we need to look at the cell dynamics very close to the wall within a region of 30 microns to the surface. And we looked at So we started to look at the cell dynamics very close to the wall, and what we did is to plot all of the trajectories of all cells which came within 30 microns of the surface, and we aligned them all at the point of closest approach. This plot here represents the recurrence probability of these trajectories, and in green we represent the mean trajectories, which shows how on average a cell scatters off a surface. To get more insight into the scattering dynamics, we can plot two additional quantities. First, we can plot the rate of change of the pitch angle, which represents how the swimming direction relative to the surface changes in time. And then we will present the rate of change of the linear velocity, which corresponds to the linear acceleration. So we plot both of them here, and we plot them with respect to times. We can observe two things. First, if we looked at theta dot, we see that around the point of closest approach, theta dot increases, which corresponds to a cell pitching away from the surface. When we look at the acceleration, we see that before the point of closest approach, it is slightly negative, corresponding to a deceleration, and after that, it is slightly positive, corresponding to an acceleration. If we look at this distribution actually a bit more closely, what we see is that not all trajectories show this fluctuation in acceleration. Here you see that a number of trajectories actually go through the scattering event with no changes in the acceleration and therefore at constant velocity. To understand what this represents for the scattering, we can plot the joint probability distribution of the maximum theta and the maximum u dot acceleration. And this is what we are plotting here. And what we find is a bimodal distribution of scattering event with a first mode which is very close to the origin and corresponds to scattering events with very small changes in the pitch rate and almost no changes in the velocity. And a second mode here where there is a more dramatic event with a stronger change in direction and more fluctuations in the velocity. This shows that two distinct scattering modes exist, and we can plot the distance to the wall at the point of minimal approach for each of these modes. If we consider the first mode here, that distribution is represented in red. What we see is that most of these events in that first mode approach a wall at a minimal distance between a normalized h of 2 or 3 corresponding to 20 to 30 micron, which means that within this first mode here, the cell did not contact the surface. If we looked at the scattering event within the second mode here, we find that conversely, the cell approached the surface to a distance less than 20 microns, and therefore we can deduce that the cell within the first mode did come into direct contact with the surface. We are by no means limited to tracking cells in the near wall region. In fact, we can track throughout the entire depth of the flow cell, so in particular as the cell depart from the surface. And what we observe is that we see many trajectories, such as these that we are plotting here, where we see that there is a repeatably tendency to return to the surface, where the cell seems to come closer to the surface and then bounce from the surface again. We can represent these trajectories in a phase space with the pitch angle in the horizontal axis and the distance to the surface in the vertical axis. And we can clearly see, this is one particular trajectory which we plot, we see an orbiting behavior of the cell. When theta, this is the pitching angle, is positive, the cell moves away from the surface and h increases, while in this half plane, when theta is negative, the cell moves towards the surface. We can characterize this orbiting behavior by representing the pitching rate in this phase space, and here we clearly see that at small distances from the wall, the pitching rate is positive, corresponding to those red values here, which means that the cell is turning away from the surface and is repelled. While at larger region, we can see that the pitching rate is positive, and this corresponds to a cell turning back towards the surface. This effect persists at quite some distances from the wall, corresponding to 10 to 20 cell diameters or 100 to 200 microns. When we plot this throughout the entire flow chamber, we can see that this effect is not only present at the bottom surface, but it is also present on the top surface. So this long-range effect is reminiscent of the hydrodynamic interaction of a force dipole with a surface, and our experiments are consistent with the existence of orbiting trajectories due to long-range hydrodynamic interactions. All right, so looking at the dynamics of motile Chlamydomonas cells interacting with surfaces, we've seen two things. First, in the near wall region, we've identified a bimodal near wall scattering dynamics. And then further away, we've identified a tendency for cells to be attracted to the surface, and therefore a long-range wall attraction. Now the question which remains is, how do these two effects influence the cell density distribution? Experimentally, we cannot isolate one effect or the other by switching them on and off. And so for this, what we did is we did simulations. We can do that in simulation, and we performed Markov chain Monte Carlo simulations to compute trajectories numerically. For these stochastic simulations, we can use our large experimental data set made out of thousands of trajectories, and we can evolve the population of cells and their position by sampling the distribution that we have just established empirically and deduce then the cell density. So before we are ready to do our simulations, there is one more thing that we need to take into account. So we've discussed the stochasticity and the distributions of a scattering event, and we've discussed the long-range interactions, but what we haven't characterized is just the swimming of a cell far away from the wall. And there is some stochasticity into that as well. And we take that into account by using all of the trajectories that we have for cells which are swimming away from the wall. And we represent the change in swimming direction by measuring the rotation rates here, omega b and omega t, and establishing their recurrence probabilities. Once we have done that, we are ready to compute our trajectories numerically by sampling the experimental distribution. And that is what we are representing here. On the bottom part here are trajectories which we're simulating by sampling the distribution, the experimental distribution. And here on the top, those are our experimental results, some of the trajectories which we have recorded. And what we see here is that both, of course, the measured and the simulated trajectories are reminiscent of a run and tumble motion, as has been reported previously in the literature for Chlamydomonas. We can see this run and tumble motion and this random walk-like motion when we plot the mean square displacement as a function of time. And we see that we have a transition from a ballistic motion at short time scales to a diffusive motion at larger time scales. In red, we represent the mean square displacement for our simulated trajectories, so for these trajectories here, and they compare well to the mean square displacement which we obtained from our experiment here in green, which validates our approach. So now that we have characterized the statistical distribution governing the dynamics of the cells, we can use our Monte Carlo simulation to evolve the position of an entire population of cells and then predict the cell density for different scenarios. So the blue symbols here represent in a log-log scale the cell density in our experiment as a function of the distance to the surface normalized by the cell diameter. We can see that there is a significant accumulation and a significant accumulation at distances which is quite significant, up to several tens of diameters, so a few hundred microns. If we now consider our simulations, which take into account the diffusive swimming behavior of the cells, and we only include the near wall scattering dynamics, meaning that we turn off the long-range effect, our simulation also predict surface accumulation, but now on a much smaller length scale, much smaller than in our experiment. We can compare these results with a simple run and tumble dynamics, and we find a similar accumulation. To test the effect of the details of the scattering dynamics on the cells, we run similar simulations, but now we assumed that we had a random scattering from the surface. And those are the red cross here, and we see that this has very little effect on the density, and here again, the simulations do not reproduce the extent of the surface accumulation observed experimentally. If we now include the long-range interaction observed in the experiment, our simulation now predicts the cell density distribution observed in our experiments closely, and in particular the extent of surface accumulation. To conclude, the large dataset collected with our experimental setup allows us to relate the surface accumulation of cells to a long-range cell-surface interaction consistent with hydrodynamic interactions. I thank you for your attention.